Hi, welcome to Chapter 15 vocab video. The first word is morphology. Morphology means the study of the shape of something. So in the context of Chapter 15, we're talking about sort of diagnosing infections. When we're looking at the morphology of a disease agent, it's like we're looking at it under the microscope to see what shape it is, oftentimes gram staining it, so just sort of physically looking at it. When we're trying to identify an organism, a lot of times we will use a series of tests and a dichotomous key. So a dichotomous key is a taxonomy tool that's used to identify specimens. So you can do this with animals. Does it have 2 legs or 4 legs? Okay, it has 4 legs. Does it have fur or no fur? Okay, or scales. And then you follow the dichotomy. It's basically like a flow chart that leads you to your final genus and species. So it's different in microbiology because we're not looking at anatomical features. We're usually looking at biochemical features, like is it gram positive or gram negative? Does it have catalase or no catalase? Does it ferment lactose or not? And so on and so forth, and you follow a flow chart until you get to the organism that you have. Another test that can be done in diagnostics, but also in biotechnology, is polymerase chain reaction, PCR. We see this one in multiple chapters because it's so commonly used. It's a reaction that basically does DNA replication in a tube so that you can make lots and lots of copies of a single piece of DNA. And sometimes we call this amplification of DNA, making lots of copies. Hybridization is also a genetic technique. PCR is a genetic technique because you're looking at the DNA. Hybridization is a technique where you use a known piece of DNA, which is called the probe, and the probe is attached to something fluorescent or an enzyme that can make a colored reaction go, and so you're probing your sample with this DNA, this known DNA, to a known organism to see if the sample tests positive for that organism. So what you're hybridizing are these basically 2 different pieces of DNA, the DNA probe and the sample DNA. So it's a DNA test to look for or to diagnose certain organisms, identify organisms. Another way that we can do diagnosis is to look at immunology. Serology is looking at the antibodies in somebody's serum. It's the study of serum and the components of serum, and antibodies are a major component of serum. And so looking at what antibodies a person makes can tell us whether they are infected with a certain organism or whether they've been infected in the past and have immunity. So serology can be used to diagnose. This is often the case with HIV. We'll see that we use a test called an Eliza, which is very common, that tests for HIV antibodies. But it can also be used in research. It's being used currently in coronavirus to look for people who have recovered from coronavirus to test whether they have immunity to it. And it's used a lot of times in people, like for example, pregnant women, to check if they have immunity to things like rubella, which is a virus that you get vaccinated for, usually in childhood, that can cause birth defects. And so when you get pregnant, you want to make sure that you won't get rubella, so they can do serology to check your antibody titers. I'm going to skip to this word here. All right. Serology can be used to check your antibody titer. Titer is just a measure of the amount and type of antibodies that you're making. So, and there's different types of antibodies that we've talked about, IgE, IgG, IgM. So testing different classes of antibodies tells doctors different things about the immune system. So titer is what we call a measure of when we're measuring antibody, kind of like antibody concentration. Another type of serological or immunological diagnostic test is an agglutination test. So an agglutination means to clump together. So antibodies cause, when they bind to antigen, remember antibodies have 2 bonding arms, they're Y-shaped. So they can bond to 2 different antigens and then basically kind of handcuff them together. And they can form these clumps of antibody-antigen clusters. And when they do that, like in a dish, we can take these microscopic antigens and antibodies, but when they all start to clump together, they become big enough for us to see. So that's what an agglutination test is. You're adding antibodies to a sample with antigen, and if it clumps, then it's positive. Or another form of agglutination test is you flip it and you add antigen to a patient's serum, and if they have matching antibodies, then it causes clumping. So if there's a match of antigen and antibody, no matter the source of those antigens and antibodies, you have an agglutination test. Another immunological test that uses antigen-antibody interactions is called an ELISA. We call it an ELISA. That's actually an acronym. It stands for Enzyme-Linked Immunosorbent Assay. So the immunosorbent part means that you are absorbing antibodies to a test well in like a plate. You could do it in tubes, but it's easier to do it in these large well plates. All right, and then you are adding a secondary antibody that has an enzyme linked to it. And that enzyme causes a colored reaction if the test is positive. That's an oversimplification, but I'll explain it better in the lecture. But no, it's an enzyme-linked immunosorbent assay. It uses antibodies and antigens to get a positive result. Another type of test that can be done, this is usually done more in research labs when you're trying to isolate and identify proteins, but it can be used in a diagnostic way and is a common test actually for HIV, though it's being sort of beat out by more convenient and rapid tests. But a Western blot is very much like gel electrophoresis of DNA, where you run pieces of DNA out on a gel, and they separate into different by their sizes into these different bands that you can see, so you get like a DNA fingerprint. A Western blot is like a protein fingerprint. It's like the same thing, except you're not looking at DNA, you're running proteins on a gel, and you're looking at the proteins. And so that's a Western blot. And like I said, HIV is one where there's Western blotting is a common diagnostic technique. So they run the patient sample out on a gel, and then they probe it with antibodies for HIV, and if they find any proteins that they match, if the antibodies bind to any of the proteins, then you have a positive. All right, the tuberculin reaction is an in vivo diagnostic test, meaning it's in living things or in a person. So in this test, it's also called a man-toe test, or are very often called a PPD test. So they take a purified protein derivative from Mycobacterium tuberculosis, which causes tuberculosis, and they inject it subcutaneously, usually in the forearm, and then wait a couple of days because that protein causes a type 4 hypersensitivity reaction, so a delayed allergic response. And if in 24 to 48 hours later, sometimes they do 72 hours later, if it's swollen enough, then that's a sign that you have been sensitized. In other words, you have been exposed to tuberculosis and may have it. So if you have a positive tuberculin reaction, then they usually follow that up with a chest x-ray. There are false positives for the tuberculin reaction. In fact, having multiple PPD tests can actually cause you to have a reaction it can sensitize you to that protein derivative. So, but it's a quick and easy way, inexpensive way to check for tuberculosis. And generally, if anyone working in healthcare has to do this at least once a year, or at least before they start their job. Microarray is, again, a genetic technique, and it's one that's used more in research labs, not as much in the field of diagnosis, but it can be used for diagnosis. And what it is it's basically like a very small, like almost like the size of a microscope slide. And then there's like little teeny dots, like hundreds of dots of DNA, of spots with DNA on it. And They're basically all DNA probes from different organisms, and you add the patient sample, and then you add, whatever, some enzyme or something that lights up if there's a match. And if whichever dot that patient sample binds to tells you which organism they have that they're infected with. So it's fairly expensive. So again, this is not usually done to diagnose individuals. It's usually done more large scale in research, but can be used for diagnosis. And then another one that's a very very sciencey and technology-based technique. You would not have a mass spectrometer in a hospital lab. They're like $1 million machines, and they're really used by chemists. They're used a lot in forensics. So like forensic science labs will use them. So what a mass spectrometer does is it helps you identify proteins. The science is complicated, and like even a little over my head. I didn't do so well in this part of chemistry. Basically, it's a machine that breaks proteins up and then like gives you a readout, and good chemists can figure out what that readout means and figure out what chemicals or what proteins are in that sample. So proteins in different bacteria are very unique. So you look for unique proteins that belong to certain bacteria or viruses, and you can identify an organism that way. So again, very, very rarely used in diagnosis of a patient, but it can be. Mass spectrometry. It identifies proteins. So that's the end of chapter 15, vocab.